Hello everyone. In this section, we'll explore advanced techniques for diagnosing viruses based on their genetic material. The loop-mediated isothermal amplification (LAMP) and recombinase polymerase amplification (RPA) assays offer faster and more user-friendly alternatives to traditional PCR. Next-generation sequencing technologies provide a powerful tool for in-depth analysis of viral genomes. CRISPR-based diagnostics hold promise for the future of rapid and specific viral detection. What is LAMP? The loop-mediated isothermal amplification (LAMP) is a nucleic acid amplification technique with a simple workflow that is used to detect DNA or RNA inputs. It was first developed by Notomi et al. LAMP takes advantage of the strand displacing properties of the BST, Bacillus stereothermophilus DNA polymerase, in conjunction with a unique primer design to allow rapid amplification of a specific sequence at one temperature, which ranges between 63 to 65 degrees centigrade. It is an ideal technique for field, point of care, or low resource settings because sophisticated molecular diagnostic equipment is not required. How does LAMP differ from PCR? First, the reaction temperature. PCR requires cycling between various temperatures to denature the DNA, anneal primers, and extend the new strand. Whereas LAMP uses one static temperature, that is 65 degree only. LAMP strand displacing polymerase eliminates the need for high temperature 95 degrees centigrade that is required for double-stranded DNA denaturation because it moves the top strand out of the way as it polymerizes. The duration of the reaction. The time frame for LAMP versus PCR is also quite different. Most PCR thermocycling protocols take upwards of 1.5 hours, particularly if incorporating real-time fluorescent measurements, while most LAMP reactions are completed in 20 to 40 minutes. Then, the primers. PCR reactions usually use two primers, whereas LAMP reactions use 4 to 6 primers. Detection. PCR products are usually detected by endpoint agarose gel electrophoresis analysis, or if it is a real-time PCR, by fluorescence measurements. LAMP can use fluorescence measurements, real or endpoint measurements, color changes, or even turbidity changes. The amplicons. The generated product in PCR is discrete, whereas single-size amplicons, whereas LAMP creates long concateners of the same repeated sequence. This is because LAMP uses 6 primers and a unique dumbbell structure capitalizes on multiple priming points. The concateners can be double or single-stranded, contain loops and branches, and can be hundreds of times longer than the amplified target sequence length. Loop-mediated isothermal amplification requires 4 primers, 2 inner primers, forward inner primer, and backward inner primer, and 2 outer primers, F3 and R3. The forward inner primer and backward inner primer each contains 2 sequences corresponding to the sense and antisense sequences of the target DNA. Additional loop primers, loop forward primer and loop backward primer, loop R, can be included, shortening the reaction time by up to 30 minutes. Why are there so many primers? A primary goal of LAMP is to detect the target sequence as quickly as possible, and the multiple primers facilitate rapid amplification and quick detection. LAMP uses 4 core primers that are necessary for amplification to occur and 2 additional loop primers that serve to accelerate the reaction. The core primers generate DNA that contains 2 regions of inverted self-complementarity. This forms self-hybridizing loop structure at both ends of the target sequence, leading to the formation of a dumbbell-like structure. The structure contains multiple opportunities for initiating synthesis, and the strand displacing BST DNA polymerase uses these priming points, resulting in rapid exponential amplification. This generates the concatenamers, long repeats of short target sequence in a concise amount of time. Mechanism of loop-mediated isothermal amplification. Initial primer annealing is initiated due to the increased temperatures of greater than 60 degrees centigrade and the addition of certain reaction additives. The 3' module of the forward inner primer FIP anneals to the partially denatured template DNA and is elongated by the DNA polymerase. The outer primer F3 anneals to the same single-stranded DNA template and is elongated by the BST polymerase. Due to its strand displacement activity, the single-stranded FIP elongation product is displaced. The FIP 5' part F1C then binds to the F1 region of the single-stranded amplicon, which leads to the formation of the terminal loop structure. Analogous to the reaction steps at the beginning, the backward inner primer anneals is elongated and is displaced by the R3 induced elongation complex. LAMP characteristic reaction intermediate with terminal loops is the basis for efficient annealing of inner primers and loop primers. Moreover, the 3' ends of this dumbbell-like structure can be elongated by the BST DNA polymerase. Exponential amplification during the isothermal LAMP reaction leads to accumulation of different size amplicons. LAMP has various detection options, including fluorescent, endpoint, visual, that is colorimetric, and lateral flow. The most commonly used are fluorescence and visual detection. Fluorescence detection. LAMP can be monitored in real time or at the endpoint via fluorescence using an intercalating dye. If monitoring in real time with intercalating dye, there is an option to include a melt curve which can help assess any non-specific amplification. For applications requiring multiplex detection, various probe-based fluorescence methods can be used. Visual detection. LAMP can also be visually monitored with pH-based colorimetric output. In a weakly buffered solution using a pH-sensitive dye, this output is particularly useful for point-of-need testing. Detection by eye is, of course, very simple. Still, the strong color change of the phenol red dye can be measured quantitatively with a smartphone camera and image analysis or by absorbance measurement in a spectrophotometer or similar device. This can be useful for higher throughput or diagnostic tests where user judgment is not required. Other non-pH based colorimetric indicators like hydroxynaphthol blue or calcium can be used with our standard LAMP mixes if samples are likely to be strongly buffered or highly variable in pH. Recombinase polymerase amplification. Recombinase polymerase amplification or RPA assay is a highly sensitive and selective isothermal amplification technique operating at 37 to 42 degrees centigrade with minimal sample preparation and is capable of amplifying as low as 1 to 10 DNA target copies in less than 20 minutes. It has been used to amplify diverse targets, including RNA, miRNA, single-stranded DNA, and double-stranded DNA from a wide variety of organisms and samples. The recombinase polymerase amplification process starts when a recombinase protein, the UBSX from T4-like bacteriophages, bind to primers in the presence of ATP and a crowding agent like a high molecular weight polyethylene glycol, forming a recombinase primer complex. The complex then interrogates double-stranded DNA, seeking a homologous sequence and promotes strand invasion by the primer at the cognate site. In order to prevent the ejection of the inserted primer by branch migration, the displaced DNA strand is stabilized by single-stranded binding proteins. Finally, the recombinase disassembles and the strand displacing DNA polymerase, like the large fragment of Bacillus subtilis polymerase I (BSU), binds to the 3' end of the primer to elongate it in the presence of DNTPs. Cyclic repetition of this process results in achievement of exponential amplification. Majority of the reports detailing endpoint detection of RPA products reported to date rely on lateral flow assays where results are obtained extremely rapidly in a visual readout format. 3 different oligonucleotides, 2 primers and a probe, and the Twist NFO kit are typically used for assay designs compatible with lateral flow strip detection. The probe is recommended to be a 46 to 52 oligonucleotide modified at the 5' end with an antigenic label, at the 3' end with a polymerase extension blocking group, and an internal A-basic nucleotide analog that substitutes one nucleotide found in the target sequence. The antigenic label is usually a carboxyfluorescein group (FAM), but others, including Alexaflor 488 or degoxygenin, are also good candidates. The A-basic nucleotide, a tetrahydrofuran residue that replaces a conventional nucleotide, is also called a DS spacer, is placed at least 30 nucleotides from the 5' end and 15 nucleotides from the 3' end. This DS spacer can be cleaved by an NFO nuclease, but only when the probe forms a double-stranded DNA. The cleavage produces a new 3' hydroxyl group in the probe, thus transforming the probe into a primer. In addition to the probe, an opposing amplification primer is labeled at the 5' end with another label, example biotin, is also required. The second primer used is a conventional primer equidirectional to the probe. The amplicon produced in the presence of the probe and 2 primers will include 2 labels on one DNA amplicon, ready to be detected in a sandwich assay format by antibodies or streptavidin. The NFO probe and lateral flow assay. The NFO probe is exchanged at the cognate site by recombinase proteins, and the NFO nuclease cleaves the probe on the THF residue. The blocking group is released and the probe is thus converted to a primer. The double-labeled amplicon obtained from the amplification is mixed with the dye loaded onto the sample pad of a lateral flow strip. The dye binds to the amplicon in the mixture pad, and the dye-amplicon mixture is captured by the antibodies immobilized on the detection line. The excess of the dye is captured by the antibodies in the control line. Next generation sequencing. Next generation sequencing, or NGS, is a massively parallel sequencing technology that offers ultra-high throughput, scalability, and speed. The technology is used to determine the order of nucleotides in entire genomes or targeted regions of DNA or RNA. Now let us look into some applications of next generation sequencing technologies in diagnostic virology. Detection of unknown viral pathogens. This means finding viruses that cause illnesses but haven't been identified before. This helps in controlling newer outbreaks. Discovery of novel viruses. This is about finding entirely new viruses not known to previously exist but have the potential to cause newer outbreaks. Detection of tumor viruses. This focuses on using NGS to identify viruses that cause tumors. Investigation of viral integration sites in host DNAs. This refers to studying viruses as to where they insert their genetic material into the host DNA. These studies help in diagnosing and treating viral infections. Characterization of the human virome. This means analyzing the entire genetic code of the virus. Epidemiology of viral infections and viral evolution. This application looks onto how viruses spread and change over time. Monitoring antiviral drug resistance. This means using NGS to track how effectively antiviral medications work or how the viruses develop resistant to certain drugs. Safety and quality control of vaccines and bioproducts. This involves using NGS to make sure vaccines and other biological products are free of infective viruses. Detection of viral mutations. This refers to identifying changes in the genetic code of a virus. Currently, three generations of DNA sequencing techniques are available. The principle of first generation techniques is the utilization of chain termination method and read length reaching 500 to 1000 base pairs. The second generation techniques, also known as the next generation techniques, include pyrosequencing, sequencing by synthesis or sequencing by ligation methods and read lengths reaching between 50 to 500 base pairs. The third generation sequencing techniques, also known as third generation sequencing technologies, perform single molecule sequencing and the read length is measured in tens of kilobases. Sanger is the first generation sequencer. Roche, Illumina, and Solid are the second generation, and SMART and Oxford Nanopore technologies emerged as third generation sequencing platforms. A typical sequencing workflow includes library preparation, sequencing the library, data analysis, and bioinformatics workflow. The construction of a sequencing library from the given sample is the initial step for each next generation sequencing and third generation sequencing applications. Depending on the application, the targeted specific DNA regions may be amplified using PCR or hybridization probes. Further, sublibraries are obtained by linking the previously acquired DNA fragments to the adapter sequences. Since these adapter sequences contain sequences specific to the sample material, that is barcodes, this stage is commonly called barcoding. Barcoding allows the sequencing of many samples in one pot. The second step includes the clonal amplification of the selected DNA fragments. The aim here is to enable solid phase amplification of DNA fragments to create a strong and detectable signal. Each DNA molecule attaches to the surfaces, beads, or flow cells and are amplified using PCR to create a series of identical clones. Third generation technology differs from the former sequencing techniques in clonal amplification, but amplification may not be necessary. Instead, all DNA in the library is sequenced simultaneously using nanopore sequencing, thus enabling a single molecule real-time sequencing. NGS often uses a version of sequencing by synthesis methods and reads as individual bases coalesce into a polymerized strand. The final step is data analysis, also known as bioinformatics. Even though they share similar workflows, each next generation sequencing and third generation sequencing platforms have their own algorithms and data analysis tools, and each step of analysis generates large amounts of complex data. Despite these differences, these tools may often be used interchangeably. Analysis can be divided into three steps. The primary analysis is the processing of the raw signals from the instrument during each sequencing cycle in numeric or Bayes matches and is in the FASTQ format. This step is also called Bayes calling. The secondary analysis involves filtering and clipping to improve quality or to remove barcodes, if any, followed by alignment of the reads to a reference genome and collating the reads for new genomes. The tertiary analysis involves interpreting the results and extracting meaningful information from the data. Viral metagenomics, which reveals the viral abundance in a wide variety of samples using next-generation sequencing technologies, is currently one of the fastest-growing scientific disciplines. Workflow for identifying viral sequences is in most common sample types. The principal steps required for sequence-based discovery from virus cultures, environmental samples, and host-associated samples like skin, gut, etc. Bifurcating arrows indicate decisions based on different detection strategies, that is, whether to exclude the cells or whether to extract nucleic acids from single viruses cells or from the entire population. The amplification step is surrounded by dashed lines to indicate that it is optional for most workflows depending on the amount of starting material and the sequencing requirements. Likewise, there are options when choosing the type of sequencing platform. There are three main strategies for sequencing viruses from the environment: virome sequencing, bulk metagenome sequencing, and single-cell sequencing. The majority of viral studies to date perform virome sequencing, which involves size filtration to enrich for the viral fraction before sequencing, similar to what is typically done before a microscopy-based analysis. Enriching for the viral fraction, the so-called virome, results in improved sequencing coverage of viruses by minimizing the number of reads wasted on the sample cellular fraction. An alternative to virome sequencing is to skip the viral enrichment step altogether and sequencing the bulk DNA or RNA found in a sample, followed by computational separation of viral and cell derived sequences. This approach greatly expands the number of samples that can be mined for viruses, as it includes metagenomics datasets collected to address other scientific objectives and is primarily responsible for the exponential growth in viral sequence databases over the last few years. Additionally, this approach allows for identification of proviruses that are integrated into the host genome, although it remains challenging to identify the sequence boundary between the virus and host and to distinguish between viable proviruses and ancient degraded remains of proviruses. Lastly, since sequences from both viral and cellular origins are produced from the same sample, this approach allows for additional analysis based on associating the viruses identified in the sample together with their presumptive hosts. The major downside of this approach is that since a majority of reads derive from cellular organisms, it is considerably more challenging to assemble low abundance viruses or viruses with large genomes. A third approach for obtaining viral sequences from environmental samples involves using a flow cytometer to isolate viruses associated with single cells or even individual viral particles. This approach usually requires an amplification step due to small amounts of nucleic acid found in the individually isolated cells or virions. Single-cell approaches can provide a very high resolution of virus-host interactions and quantify and characterize the dynamics of the interaction. Example, the number of live tick versus lysogenic infections in a community. Single-cell virus approaches are especially useful in addressing some of the challenges of reconstructing complete viral genomes from metagenomes. A variation on single-cell sequencing involves combining fluorescently stained viral particles with either cultured bacterial hosts or even uncultured hosts from the environment and then sorting and collecting the cells that viruses target. However, with all single-cell approaches, DNA amplification can result in highly fragmented assemblies, and it can also be challenging to discriminate between viruses that were attached to the cell and viruses integrated into the host genome. Strengths and limitations of next-generation sequencing. The major strengths of next-generation sequencing is that this method can detect abnormalities across the entire genome in case of whole genome sequencing. It can detect substitutions, deletions, insertions, duplications, copy number changes in gene and exons, and chromosome inversions or translocations. It Another major strength of next generation sequencing is that it can detect all of these abnormalities using less DNA than required for traditional methods. The time to perform most next generation sequencing methods and receive results has been greatly reduced. Starting from the day the laboratory receives the specimen, it takes approximately 10 days for a physician to receive a whole genome sequencing report. Costs of sequencing the whole genome have decreased significantly over the last decade. In 2006, the cost was approximately 20 million to $25 million for sequencing a human genome. In 2016, the cost to sequence a human genome is generally less than $1,000. Limitations of next generation sequencing. There are several limitations to using next generation sequencing. Next generation sequencing provides information on a number of molecular aberrations. However, for many of the identified abnormalities, the clinical significance is currently unknown. It also requires significant bioinformatics systems, fast data processing, and large data storage capabilities, which can be costly. Although many institutions may have the ability to purchase next generation sequencing equipment, many lack the computational resources and staffing to analyze and clinically interpret the data. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR is an ancient bacterial immune system that harbors a programmable protein, Cas, that can cut DNA or RNA. The system can be repurposed as a biological search engine because it uses an RNA molecule that guides the Cas protein to search through billions of sequences to find a matching nucleic acid target. The CRISPR technology has revolutionized the field of diagnostics. CRISPR systems can be engineered to target virtually any genetic sequence, enabling customization of various diagnostic needs. The precise targeting mechanism can lead to highly specific and sensitive detection of pathogens or genetic abnormalities. What is Cas13 and what can it do? Cas13 is a CRISPR protein that cuts RNA. Cas13 finds its way to an RNA sequence with the help of a guide RNA, a programmable 30 base string of nucleotides that are complementary to those in the targeted area in the RNA strand. Cas13 binds and cleaves additional RNAs regardless of homology. This is referred to as collateral cleavage. It is this property of Cas13a that opens up the possibility of using it as a diagnostic tool. CRISPR-based diagnostics for viral nucleic acids. In the absence of the nucleic acid target, the Cas nuclease is inactive. When binding to its guide CRISPR RNA to a related target, the nuclease is activated, leading to catalytic cleavage of off-target nucleic acids. This collateral nuclease activity is turned into an amplified signal by providing reporter probes with a fluorophore linked to a quencher by a short oligonucleotide. Sherlock. How does Sherlock work? Sherlock stands for Specific High Sensitivity Enzymatic Reporter Unlocking. Cas13 can be programmed with the CRISPR RNA to target a single-stranded RNA of interest. For example, a sequence specific to a virus. Once Cas13 recognizes and binds to the programmed sequence, it can cleave surrounding single-stranded RNA molecules. In Sherlock, a quenched fluorescent single-stranded RNA reporter is added to the reaction. Cleavage of the quenchable fluorescent RNA by the activated Cas13 produces a quantifiable signal that indicates the presence of the targeted nucleic acid. To increase the sensitivity of the assay, the targeted DNA or RNA in the sample is first amplified using recombinase polymerase amplification or reverse transcriptase recombinase polymerase amplification respectively. RPA is coupled with T7 transcription to convert the amplified DNA to RNA for subsequent detection by Cas13. This amplification step, in combination with the single-stranded reporter RNA, enables Sherlock to detect DNA or RNA with attomolar sensitivity and single base pair mismatch specificity. Sherlock version 2. Sherlock had few limitations when it was first introduced in 2017, so further modifications were made to it to improve its detection system. Sherlock version 2 uses far less primer in the pre-amplification step, allowing for greater quantification without compromising sensitivity. Sherlock version 2 was adapted so that a cleared reporter could be detected on commercial lateral flow strips similar to the pregnancy tests. With lateral flow strips, the presence of your DNA or RNA of interest within a given sample is simply determined by the number of bands present on the strip. This type of readout allows for nucleic acid detection almost anywhere, as lateral flow strips are easy to transport and work rapidly, providing reliable results in less than an hour. Detector. Detector stands for DNA endonuclease targeted CRISPR trans reporter. It utilizes Cas12a, which cleaves double-stranded DNA and activates non-specific cleavage of single-stranded DNA. They use the system to detect carcinoma associated with human papilloma virus from clinical specimens, also using RPA with attomolar sensitivity. Detector works similarly to Sherlock. The Cas12a is targeted to a specific DNA sequence, such as a human papilloma virus genome, via a CRISPR RNA. As an single-stranded fluorescently quenched reporter, which will produce a signal when single-stranded DNA is degraded, is added to the reaction. To enhance sensitivity, the DNA is first amplified through isothermal amplification by RPA. When Cas12 CRISPR RNA base pairs with the DNA of interest, the DNase activity of Cas12a is initiated. Surrounding trans single-stranded DNA, including the single-stranded DNA FQ reporter, are subsequently degraded at the rate of 1,250 cuts per second. A quantifiable fluorescent signal designates the presence of DNA of interest, in this case, the human papilloma virus. Advantages of CRISPR-based diagnostics over PCR methods. High specificity. CRISPR precisely targets sequences due to enzymatic recognition. Signal amplifications leads to quicker turnaround times and therefore faster results can be obtained. Isothermal reactions enable compatibility with simpler, cheaper tools, which do not require complicated and costly equipments. Therefore, it is a low cost and effective technology. Programmable system allows for swift test creation. Future possibilities of CRISPR diagnostics. Easy to use testing kits for various applications may be developed. Benchtop integration. Compatibility with existing lab equipment would help in getting quicker diagnostic results. User-friendly tools could be developed for on-site diagnostics. There is a lot of potential for developing at-home testing kits for self-administered tests, but with considerations. What are some of the challenges of at-home CRISPR diagnostics? One is the data privacy. Ensuring user information remains secure is one of the challenges. Further, there may be a need for guidance after receiving test results. There are ongoing discussions on responsible use and support of CRISPR-based diagnostic kits. These are few references for further reading. Now let us discuss some MCQs on this topic. Which of the following diagnostic techniques amplifies viral nucleic acid at a constant temperature? A. PCR, polymerase chain reaction. B. LAM, loop-mediated isothermal amplification. C. NGS, next generation sequencing technology. Or D. ELISA, enzyme-linked immunosorbent assay. The right answer is B. LAM, loop-mediated isothermal amplification assay. CRISPR-Cas systems are used in viral diagnostics because they can A. Amplify viral RNA efficiently. B. Cleave specific target sequences with high accuracy. C. Grow viruses in a laboratory setting. D. Stain viral particles for visualization. Answer is B. Cleave specific target sequences with high accuracy. NGS or next generation sequencing offers the advantage of A. Detecting only known viruses. B. Identifying novel viral strains. C. Fast turnaround time for results. D. Being a simple point of care test. Answer is identifying novel viral strains. RPA kits are designed to be A. Universal for all types of viruses. B. Specific for a particular viral target. C. Time consuming to develop and validate. D. Inaccurate for point of care applications. Answer is B. Specific for a particular viral target. Thank you for your attention.